Okay, so today talking about, I changed my title a little bit. I called it Toxic Microcystis Blooms in a Low Nutrient Lake. Some insights from high resolution monitoring. So high resolution through time, high resolution through space as well. And I have this photo here of our field tech, Clark, holding this gravity core. And really this whole talk, not this whole talk, but one of the central themes is the interaction between the sediments, the muck at the bottom of the lake on the lake bed, and the water column. And you can see that in that photo there. And it's Algae Day. And I mean, we're not really celebrating algae, are we? We're trying to figure out how to make them go away. But here's this nice heart-shaped Microcystis colony that I saw from Lake Eugenia. So maybe it was a gesture of goodwill or something from the phytoplankton. Okay, so we'll start at the beginning. What do algae need? And I put algae in quotation marks. I've got to be, one of my colleagues is always telling me I'm pedantic. What algae, we're really talking about cyanobacteria, right? They're not algae, they're bacteria, and they share characteristics with algae. They get their energy from the light. So what do they need? Well, they basically need the same things your house plant needs. They need water, and they've got lots of that. Of course, carbon dioxide, the water's full of it, and they need light, and they need nutrients. Light's not usually limiting for them. It sometimes can be, and I think it gets ignored a little too much maybe in lake management studies, and so I'm going to talk a bit about light today. But it really usually it's nutrients that are limiting phytoplankton, cyanobacteria biomass, and it's usually phosphorus that's in shortest supply relative to demand. So back in the old days, 1960s, Lake Erie's turning green. It wasn't a simple matter to figure it out at that time, but now looking back, I mean, this was the paradigm. You've got really high phosphorus loading to the lake, concentrations go up in the lake, cyanobacteria tend to outcompete the algae, the other phytoplankton, when phosphorus concentrations are high. There's different reasons they might do that, but one is if there's enough phosphorus, nitrogen tends to become limiting, right? There's so much phosphorus, it's no longer scarce relative to demand. Now they need more nitrogen. And cyanobacteria, some of them, can take that nitrogen from the air. So we're all breathing nitrogen right now. We're not using it, but almost 80% of the air is nitrogen. And so they can use that nitrogen and outcompete the algae that can't. One of the theories anyway. Okay, so, but that doesn't explain a lake like Hutchinson Lake here, which gets used as an example of an oligotrophic lake that experiences or has experienced a severe algal bloom fairly often. So you may have heard of it, but it's smack dab in the middle of Algonquin Park. This is not a situation in which there's a wastewater treatment plant discharging into the lake. There's not intensive agriculture around the lake, nothing like that. So why did Dixon Lake have this really extreme algal bloom when it has a shoreline that looks like what you can see pictured there, with no development, no point sources, no agriculture, nothing to supply phosphorus to the lake? Well, the thinking at the time, and maybe still is that it's climate change, right? If the nutrient loading and other things haven't changed, well, we know the climate is changing, so perhaps that's responsible. Here's a graph from a paper from some Ministry of Environment scientists showing, and you may have seen this before, but showing an increase, not in the number of algal blooms, but in reports of algal blooms for the first time in that water body. So the idea being here that there's a lot of lakes that haven't experienced a bloom before that are experiencing their first bloom, and that tends to be increasing through time, which suggests perhaps a climate angle. I think we have to be realistic and realize these are reports of blooms too, right? Not quite the same as actual blooms. These are reports. And we know another thing that's gone up quite a bit over the last few decades, and that's residential property values. So maybe you care more about your property, you're more apt to report a bloom. So we have to view something like this with a bit of scrutiny, but it does seem that the evidence is blooms are getting more common and more severe and happening in lakes that they didn't used to happen in before. So what's going on? I realize Lake Eugenia is not in Muskoka. I realize this is the Muskoka Watershed Council. Lake Eugenia, I think, has a lot in common with a lot of the Muskoka lakes. So it's a small lake. Nutrient levels are pretty low. It's clear. And it has, at times, it can have these Microcystis blooms. So the lake, you can see the map there. It's located kind of in the middle of nowhere, really, west of Barrie and south of, Collingwood, Meaford kind of area. So there's a bit of agriculture in the catchment, but it's not, the nutrient loading to the lake is not that high. But they have these late summer and fall Microcystis blooms that are toxic. The lake was created, I should say, over 100 years ago for power generation. So it's actually a naturalized reservoir, but it's very lake-like. And you can see the bathymetry there at the bottom of the screen. So it's a pretty shallow lake. Most of the lake is 1 to 3 meters deep. So it's pretty shallow, except for this deep area, which is, depends on the water level, it's a reservoir, but 10 to 11 meters in this deep area. And that's really what I'm going to focus on today in this talk. Okay, so that slide I had about the old paradigm, high phosphorus loading, high phosphorus concentrations, algal blooms. Simple enough, right? Well, okay, so Lake Eugenia has these toxic Microcystis blooms. The phosphorus concentrations in the spring, based on the Lake Partner Program, and I'm assuming some people are familiar with that program, maybe partake in it. You can see the plot there. They've been consistently below 10 micrograms per liter. So 10 micrograms per liter is the Ministry of Environment's old provincial water quality objective for a high level of aesthetic protection. It has been consistently below that concentration, at least in the spring. The other graph here showing you a seasonal trend in the phosphorus. So it tends to increase. That blue line there is the phosphorus increasing during the ice-free season, during the open water season. So something's going on there. Low concentrations in the spring, still low, but increasing a bit later in the year. Okay, so if it's not the phosphorus, or at least not in a simple, direct way, what might it be? So we'll talk a little limnology 101 here, a little bit about lake stratification. You might already know this, you might not. But, I mean, if you live on the lake or spend time by the lake, you know that there's a lot of mixing going on early in the year as well as late in the year, right? It's cold, strong wind. So the lake, especially in shallower systems, but pretty much any lake, is going to be mixing right down to the bottom, okay, vertically. The wind is mixing the water column completely. Later in the year, it warms up. That top layer of the lake, we call it the epilimnion, that surface mixed layer. It warms up. You get a temperature gradient at depth, maybe at 5 or 10 meters down. And this denser, colder layer of water at the bottom becomes isolated from that top layer. So we call that the hypolimnion, that layer at the bottom. Okay, so stratification, strata, layers. So they're physically isolated, and because of the physical isolation of these two layers of water, the chemistry of these layers can change. So eventually what happens is the oxygen can get used up in the bottom layer. It's getting used, but it's not getting produced, right? There's not a lot of light down there. There's not a lot of photosynthesis. And once that oxygen's gone, we get chemical changes. So phosphorus and other things like iron and manganese coming out of the sediments. So the sediments, that layer of muck on the lake bed at the bottom there. Okay, we call this internal loading of phosphorus and other things. So external loading, it's coming from the watershed, it's coming from rivers and things like that. And this is internal, it's coming from the bottom of the lake. So that was one of the things we wanted to look at in Lake Eugenia. So this was last year, we did this work. We had a bunch of sensors that we put at the deep spot by the dam, and then we had 2 moorings of oxygen, temperature, and conductivity, which is our tracer for ions. We had these loggers at shallow sites as well, so 3 sites throughout the lake. You can, visit a lake once and learn a lot about it just from sampling it that one time in the spring. You could sample monthly or even bi-weekly throughout the season and learn considerably more. But there are things that are happening on more of an hourly to daily time scale that kind of sampling, you're just going to miss it. And so for those situations, you really need to have loggers, some kind of sensor, something automated that can be measuring things when you're not. So we had these sensor strings in the lake. We just did like normal water quality sampling as well from the top and bottom of the water column, and did some sediment profiling. So there's that same photo of our tech Clark there with a core taken from the bottom of the lake. And this was kind of a 2-part project. So we also had a citizen science, I guess you can call it, angle, where the residents were reporting blooms, so taking photos and also taking samples that I could look at under the microscope. Again, because we can't be there all the time, and there's no real predicting when these blooms are going to happen. So this was really valuable information, just to have the basic information: where did the bloom happen, when, roughly how big was it, and here's some photos and a sample. Okay, a whole bunch of boring numbers. The point of this large table of boring numbers is to show you that for all these parameters, these different chemicals that we measured in the lake, the maximum values measured in 2023, none of them were above the provincial water quality objectives. Okay? And this is a lake with toxic cyanobacterial blooms reoccurring on a fairly regular basis. Okay, I mentioned light earlier. The image of the Secchi disk might be familiar to some of you or not, but it's a disk. You lower it in the water, the depth at which it disappears, we call that the Secchi depth. So it's just a simple way of measuring the transparency, clarity of the water. So this gets measured a lot, and here we wanted to do a bit with the data and ask the question, where in the lake can the light reach all the way to the lake bed, to the bottom of the lake? Enough light that it could support something photosynthetic like algae, cyanobacteria. Okay, that's the question. So we call that the euphotic depth. And basically what the data are showing, that chart there shows you, is that up until August, the deepest part of the lake that was 11 meters roughly deep, there was enough light at the bottom of the lake to support net photosynthesis, net primary production, I should say. So to support the growth of algae or cyanobacteria on the bottom of the lake. Okay, because remember what algae need? They need CO2 and water, which they always have in a lake. They also need light, and they need nutrients. Okay, so nutrients. So phosphorus. I already said that the phosphorus concentrations in the lake, in the water column, are actually pretty low. Usually in the spring, at least, they're below 10. They never made it above 20 micrograms per liter at any time, which is relatively low for a lake with problems with cyanobacteria. Okay, so we're coring the sediments in the deep part and the shallow parts of the lake to look at how much phosphorus is there. Okay, not always the most useful thing to know, because generally the sediments are always pretty enriched with phosphorus. Like if you've dug up the sediments from a lake, you know this is a bunch of muck, right? There's lots of nutrients and inorganic and organic matter there. The question really comes down to, can that be released? Doesn't matter if it's there, if it can't be released and accessed by the algae and cyanobacteria. So we look at what it's bound to. Well, we didn't. We sent this to a lab at Guelph University. And based on what the phosphorus is bound to, you can infer, is it releasable or not? And depending on the site, something between 1/3 and 1/2 of all the phosphorus is potentially, or was potentially releasable. And we did this early in the year before any of it would have been released, so in May. Okay, so there's a bunch of potentially releasable phosphorus. Now the question is, are the conditions right for that phosphorus to be released? Whole bunch of graphs, a lot going on here. I understand, and I apologize. But if I can sum it up at a high level, we wanted to look at, are these shallow areas, 2 to 3 meters, so up to maybe 10 feet at most in depth, are those stratifying? Are those separating into layers where the bottom might lose its oxygen and that phosphorus is coming out of the sediments? The assumption usually is no, it's too shallow, right? The wind energy is going to mix that right to the bottom. So this is our high frequency data looking at, in August when it's really hot and when there's those periods, we got, I think, more now where the wind is really low for maybe 2 or 3 days at a time, is it stratifying. And the top panels there show you the temperature difference between the top and the bottom of the water column. And that, yes, the water column was stratifying for, it's dynamic. It was stratifying for, hours to days and then destratifying, and that happened through the whole season. The middle panels are showing you that the oxygen concentrations, except for a little bit in the blue time series there for the site LE5, the oxygen didn't get used up. So I was really hoping for this interesting study here that we'd see all this internal loading happening in the shallows, but the evidence is that even though it was stratifying, the sediments aren't using up all the oxygen above them quick enough for this internal loading to be an issue in these shallow areas. But it's something certainly, I've seen it in other lakes where a few hours, there's so much oxygen demand, so much organic matter in the sediments that oxygen gets pulled down within a day. And the internal loading happens fairly soon thereafter. That didn't happen here. Okay, so then the question is, what's going on in this deep area of the lake? Again, it's a little over 30 feet, 35 feet, 10 meters, something like that, depending on the location. What's happening there? Again, apologies for another sciencey, information-heavy graph here, but I don't see any way around it. So the blue here, we're looking at the dissolved oxygen in the hypolimnion. So that's that bottom layer of the lake. And the green is the conductivity. So that's a measure of the ions in the water. And we see a couple things. We see the oxygen is declining in the hypolimnion, which we'd expect, and it gets used up by early August, completely gone. Before that, you can see those vertical blue-green lines, and those are indicative of when the residents were reporting algal blooms. Okay, so those started shortly after the oxygen started to decline, and there were these spikes you can see in conductivity. Something's happening, either release of nutrients or possibly even algae moving throughout the water column, and there's substances associated with their colonies that might be picked up by the sensors. Not entirely sure here. I should say our sensors was 2 meters off the bottom. So I think the internal loading is happening. The oxygen, we're still measuring oxygen because we didn't put these things right down in the sediments. We didn't want the sensors to get fouled by the muck. So we had them roughly 6 feet off the bottom. So, okay. Next slide. So what's happening with the chemistry? And here the orange time series represents the oxygen, same as in the last slide. It's just showing it goes down and eventually gets used up in August. So that bottom layer of the lake is anoxic or devoid of oxygen. Before that happens completely, you can see the top there is the phosphorus concentrations, and that blue line shows you the phosphorus concentrations near the bottom of the lake started to go up right before we started to get reports of blooms from the residents. At that same time, the bottom chart there, it's showing you the same information as the top chart, except for that dark blue time series. That's cyanobacteria biomass actually measured by looking under a microscope and counting the cells. So it's not just people's reports that there was a bloom. We're saying how much cyanobacteria are actually, blue-green algae, are actually in the water column. So you can see that as the oxygen gets used up, phosphorus increases, cyanobacteria biomass increases. If you look at that orange time series, there's a weird kind of wiggle there. See how much variation I don't know how to point with this pointer at the, okay, so it's this area at the very beginning where the orange starts. See how it's really wavy there? Where it says the word logger on the bottom. Anyway, I think everyone knows what I'm talking about. Yeah, exactly, right there. There's a lot of variation, a lot of wobble there in the oxygen. So just something to note. I'm going to come back to that in a subsequent slide. Okay, so I mentioned we had a taxonomist, a professional who just does this all day, every day, look at the samples and tell us what's in them. So you can see that this is the amount of phytoplankton there was in the water column. Early in the season, it's a mixed community, lots of different types of algae, diatoms, things like that. And then by July, it's almost all cyanobacteria. That's that blue-green color there. And then in August and September, just very abundant. They basically took over the whole lake. There was nothing in the water column other than Microcystis, which is this genus that, I mean, it's one of the most common bloom-forming taxa that there is, and it produces a number of toxins such as microcystin. Okay, so vertically in the water column, again, at this deep site, this is a measure, this green line, how much phytoplankton is in the water. This is chlorophyll A estimated by fluorescence, but basically how much phytoplankton is in the water column at different depths. If you look at June 20th there, so the middle plot in the top row, see there's a big spike in the chlorophyll. Yep. Just off the bottom at about 9 meters, right where it says FP chlorophyll A there. So there's this big concentration of something that fluoresces, like phytoplankton, down at 9 meters depth, so way down, almost 30 feet, if not 30 feet depth. So what might that be? Okay, the most complex slide. Again, I apologize, but go through it. But it gets better after here, I think. Just couldn't help myself. Okay, so I mentioned this oxygen that was really wobbling, right? And you can see it in the red box there. If we zoom in on that, there's a trend, it's going down. So bottom of the lake, the oxygen's going down, but it's wobbling like crazy. So let's remove that trend of decrease over time and just look at the hourly trend that remains. That's that center graph there where it says look at hourly trend. And it still kind of looks like just like a shotgun blast. There's no real pattern that you can see. But if you zoom way in, if you do a locally weighted regression, so basically a little smooth curve that captures the variation, and we zoom in on that, now the bottom right graph, all of a sudden, there's a very clear pattern. That's that red line that's kind of looks like a jump you'd go off downhill skiing or something. So what that is, that's a pretty characteristic pattern that you see wherever there's plants or algae in a lake or river, and that's based on the time of day. So that declining trend overnight, the oxygen keeps going down. Everything's breathing, just like we are. I trap you in a room and you keep breathing and it's airtight, you're going to use up all the oxygen, right? So the oxygen's going down overnight when it's dark, and then around 10 o'clock there, you can see the trend reverses. It's getting bright outside. The phytoplankton are now producing oxygen, photosynthesis, oxygen's going up. So this, what we call a diel, or 24-hour pattern in oxygen, is representative. Well, it tells us something's down there doing photosynthesis. So this spike at 9 meters is not, dead matter that's fluorescing. These guys are alive down there, and they're actively growing at 9 meters depth in the hypolimnion. It's very faint, but if you tease it apart, it's there. Which then leads us to ask, well, why on earth are cyanobacteria way down at 9 meters, way down below the mixed layer where they belong? Well, we have to think a bit about their life cycle. So I don't want to make any assumptions about the group of people here, but I'm going to guess at least half of the people in this room go to Florida every, think it'd be a good guess? No? So the cyanobacteria, in a comparable way, every winter, the cyanobacteria go down to the sediments and they basically overwinter. They become inactive and they wait out the winter, basically sitting on the muck at the bottom of the lake. This is not exactly a technical explanation of what's happening, but it's essentially correct. They're kind of like going into hibernation for the winter. Next season, ice is gone, the sun comes out, it's the open water season, they're going to become active again and start metabolizing again. So they're starting their season down there already. They're starting the year on the sediment surface, arising from their slumber. And as I said, remember that Secchi disk slide, there's enough light down there at the lake bed already. So there's enough light. We know the phosphorus is in the sediments. We know it's releasable, or at least roughly a third or half of it is. So there's quite a bit of releasable phosphorus. The conditions are right. We saw that it's being released. So if you're a cyanobacterial cell, I think you're happy, right? You've got light, you've got nutrients. You don't really need to go anywhere until things change. You can grow way down there at depth. And if we think about a bloom like the one that happened on Dixon Lake, or that's happened on countless oligotrophic lakes and other lakes that are more nutrient enriched, but regardless, you see these situations where everything is clear and then all of a sudden overnight there's this bloom that appears out of nowhere. How can that possibly happen, right? Like if you leave some fridge out of the refrigerator and you forget about it, go to bed and wake up the next day, is it just like all mold? No, but if you left it there for a week or two, it would be, right? So the generation time of the phytoplankton is variable, but it's something like maybe 1 to 5 days. So how could this happen overnight, where we go from a situation where the water's all clear to a situation where it's pea soup? Getting a little speculative here, but just, good old logic. I think it's already there. The bloom is already there. It's associated with the sediments, and it's becoming buoyant and entering the mixed layer. Now all of a sudden we see it as a bloom. The other explanation, you could just have it really, if it's really deep, the cells are distributed throughout the water column, they get concentrated. But either way, that biomass already has to be in the lake. And when we say, oh, there's a bloom all of a sudden, we just mean it's been concentrated, it's visible to us, it's interacting with the surface of the lake. Okay, so speaking of blooms here, so these are some photos the residents took of the blooms. They started out kind of as these like ring around the bathtub type blooms, where the lake was generally clear and there was just a little kind of fringe of pea soup-like material. And then later in the season, it was, essentially the whole lake went green. A little disappointing, I guess, when you're trying to make sense of this. There wasn't really any pattern when we look at this map here showing all the reports we got from the residents. There wasn't any kind of clear pattern, they started in this part of the lake and they moved throughout. Or if you look, you can stare at this all day, and there's no real, where there were people who were participating, there were bloom reports. So we can't do this in a relatively short study, but if you had a lot of time and a lot of money, you could do something like the hydrodynamic modeling to figure out, okay, let's assume the bloom started at the bottom left here by the dam. In this deep part, could we model the dispersion based on currents and wind and everything throughout the lake? But that's an extremely complex exercise. Okay, so I mentioned the old paradigm, essentially, that where you have high phosphorus loading, cyanobacteria are going to come to dominate the community. So maybe the new paradigm for these oligotrophic or low nutrient lakes, where phosphorus loading is not high, has more to do with weather and the physics of the water column. So higher temperatures, more stratification, the phytoplankton have more access to phosphorus from the sediments, and we get more blooms. Okay, so after all this theoretical and empirical data, but possibly a little abstract at times, you might be asking, you know, okay, so what? So what does this mean? How do we manage these blooms? If they're happening and you think you understand a possible mechanism, what does this mean for making them go away? Not to say that we don't need best management practices like making sure everyone's septic systems are working, making sure sources of nutrients are well set back from the water's edge. If people could, you know, keep their shoreline natural, that would be nice. These things are all necessary, but I don't think they're always sufficient. I mean, again, if you look at a lake in the middle of Algonquin Park, nobody's mowing the lawns in there. There's no septic systems. So I think necessary, but not always sufficient. I think there comes a time, if you, no, you don't have to do anything, but if you want to stop the blooms, I think there comes a time when you have to actually stop the transfer of the phosphorus from the sediments to the cyanobacteria. Okay, so for this study of Lake Eugenia, we evaluated a number of potential management options. I'm not going to go into depth in all of them here. We identified Phoslock, which is this phosphorus binding chemical, as the best option. It's unfortunately been misclassified as a pesticide by the federal government. It cannot be imported into Canada. So we identified alum as the best available option. Alum is aluminum sulfate. It's used in wastewater treatment and has been for a very long time. So it binds phosphorus and keeps it from being released under anoxic conditions when that oxygen gets used up. So our recommendation in this study was to treat that deepest part of the lake, which you can see there in red, inject this into the hypolimnion, the bottom layer of the lake. Nothing alive is in there, it's anoxic, there's no oxygen. So concerns, I mean, the concerns around the things already living there are reasonable, right? But there is nothing living in that part of the lake, and if this is buffered, it should have no effect on the pH and no toxicological effects that are major. And it's, to start with anyway, small area of the lake. It's less than 1% of the area of the lake, so just that deep zone. So that was our recommendation for that lake. So alum, again, it's aluminum sulfate. I understand, I think generally the perception might be, okay, so what you're saying is you want to inject a chemical into the lake at a large scale. I don't like the sound of that. Understandable. I mean, it's been used for a long time in the United States on hundreds, if not thousands of lakes. So there's a lot of peer-reviewed literature on it. You can buy it at the grocery store. As you can see there in this photo, that little orange jar, that's alum. So it's used in baking. And here I did a little tinkering at home with just some liquid plant food to get the nutrients and alum to bind it up. And you can see on the other side of the screen there, that little mason jar. So that's what it looks like after the flocculation has happened. So the chemical reacts with the phosphorus and other things in the water, and it forms a precipitate that forms this kind of like snowy layer on the bottom of the lake. And are you able to play the video? I'm not sure if this will work or not, but one of the concerns in a shallower lake like Eugenia with a relatively fast flushing time is, I mean, we were upfront about this, you just have to be aware that if it gets stirred up at a time when the lake is draining, it's going to get flushed from the system. So I think people tend to worry about toxicological effects, which there isn't a lot of support that that's a major concern if it's applied properly. But another concern, of course, if you're paying for something is, you know, I don't want to put it in there and it just gets flushed out of the lake. So that's another concern, is the longevity of the treatment, and it can be really, really variable depending on the particular characteristics of the lake. So with Eugenia, it's a reservoir. it's drawn down, has a pretty fast flushing time on an annual scale, but it's really just drawn down at the end of the year, and it's not drawn down much, if at all, during the summer. So we don't think it would be an issue in the short term, but it's a source of uncertainty, I guess, for that lake. So it does get stirred up, but as you can see, the flock kind of quickly settles back out of the water. Phosphlock is a lot denser, that other phosphorus-binding chemical. It has a much higher density. It's a clay, bentonite clay, and so it doesn't get resuspended as easily, and that was our rationale for saying it would be preferable over alum. Okay, final thoughts. So summing up, I've got the little movie poster here from What Lies Beneath. So it's really, I think just looking at, water column spring total phosphorus, that is an extremely valuable piece of information. I don't know where we'd be without the Lake Partner Program, but it's not the whole story, and I think sometimes you have to look at what's happening down deep and through time and ask questions like, where does light reach the lake bed? What's going on with the sediments in terms of phosphorus loading? And of course, recognizing that the cyanobacterial resting cells are hanging out in those very same sediments. The long-term prognosis, which I guess the next talk is about, the future of algal blooms, sorry, but doesn't look great. Warmer air, less ice cover. That means earlier stratification. That means stronger stratification. That means more oxygen depletion. And this whole mechanism is just going to get exacerbated with climate change. I mean, it already is it's already happening, but I think it's just going to get worse. So then the question is, maybe it's time for more of an active lake management approach in Ontario. Maybe not. Maybe it'll be another decade or two. I don't know. But just throwing it out there. And I'd like to thank all these people who funded our study and helped provide input. And that is all. I'll take any questions. Thank you.